decent feedback.
I can tell you in a minute if I'm showing up. I can't until I've
Well,
got
give
it
me
in.
some feedback as to what's going on.
I'm just restarting the meeting. I'm there right now. Dad, I'm there right now. Whether or not I'll stay there, I don't know, but I'm there right now.
Does that mean your camera's working?
I think so. Right now it is.
We see you.
Okay. All right, Dad, it's working.
All right, bye.
I love you, Dad. Okay.
Thanks, Dad.
Yeah, thanks, Dad.
Oh, he's already hung up. He only can handle me in small doses sometimes. All right, let's jump into this again. So looking at this Henry's Law, I have a beaker. This is even worse than the last beaker and it's drawing, but I have a beaker and I introduce into this beaker carbon dioxide that has that's not what I meant. That has a partial pressure of, let's say 50 millimeters of mercury. And I have control copy. And then I have a oxygen that has a partial pressure of 75 millimeters of mercury. So I introduce them both in, and just like everything else that we talk about, it's going to move down. Each of these gases is going to move according to its concentration gradient. So initially within the water, the partial pressure is 0 for both of them. But within a few minutes or however long it takes, they're going to equilibrate. And the partial pressure in the water for carbon dioxide is going to be 50 millimeters of mercury, when initially it was 0. And 75 for oxygen. Nevertheless, the solubility of carbon dioxide is going to be higher. It's going to be more easily solubilized into water than oxygen is. So it's going to have a solubility of about 2.5 millimeters of mercury. Not 2.5 millimeters, millimoles per liter. And oxygen gas, on the other hand, is going to be much lower, for example, 0.025 millimoles per liter. So they each move according to their own concentration gradient. And carbon dioxide is much more soluble in water than oxygen is. So then. Oh, wrong one. With this in mind. So that was Henry's law. That they're going to dissolve in liquids in proportion to their own partial pressure, their own individual partial pressure. And the solubility of gases in liquids differs. With, for example, carbon dioxide is much more soluble than oxygen is. What does this have to do with the respiratory system, and why does it matter? When we're looking at gas exchanges, air that's coming into the lungs has a partial pressure of oxygen of 159 millimeters of mercury. Remember how you'd get that? So 20.9%, oxygen is 20.9% of atmospheric air. And thus, if you take 20.9, excuse me, 0.209 times 760 gives you 159 millimeters of mercury. Carbon dioxide, its percentage in the air is 0.04%. So 760 times 0.04% gives you 0.3 millimeters of mercury. So this is the air that's coming into the body. So it comes in, it gets dissolved in the water within the lungs. And remember, that's going to drop because oxygen is not very soluble in water. Its partial pressure is going to drop. Carbon dioxide is much more soluble in the lungs, in water. And therefore its pressure, partial pressure is going to go up in comparison to the outside air. So it comes in, it circulates, it moves into the blood. It's going to equilibrate between the alveoli and the surrounding capillaries. And so it's going to equilibrate until the partial pressures within the blood and within the alveoli are approximately the same. So once it's in the capillaries and it leaves the lungs and enters the heart, the left side of the heart, then gets pumped out into the systemic capillaries. It has a partial pressure of 9. Oxygen has a partial pressure of 95, and carbon dioxide has a partial pressure of 40. It then starts to flow throughout the systemic tissues. And in the tissue, interstitial fluids and tissues, the partial pressure of oxygen is 40 millimeters of mercury, and the partial pressure of carbon dioxide is 46. So you can see it's dropped. So what happens is that oxygen is going to unload. It's going to go from 95 to 40. And the carbon dioxide levels are going to go from 46 to 46. Am I making sense here? I can only see one face.
Yes,
Okay.
I'm here.
Yeah, it makes sense.
Okay.
Yep.
So it starts 95. It comes down. The blood is coming down. And the blood still has this partial pressure of 95. It comes down and it's got, so the blood has a partial pressure of 95, has a partial pressure of carbon dioxide of 40. As it comes down into the interstitial tissues, it's going to flow from the blood into the tissues because the partial pressure of the oxygen is higher in the blood. Everything always moves from a high to a low. Carbon dioxide, on the other hand, has a higher partial pressure in the tissues than it does in the blood. And so it's going to flow into the blood until the blood has the same partial pressure as that of the tissues. So in the lungs, with gas exchange, oxygen gets loaded, carbon dioxide gets unloaded according to their partial pressures. Oxygen gets carried from the alveoli into the systemic tissues. Carbon dioxide gets carried from the systemic tissues to the alveoli. And with the tissues, the exchange, oxygen is going to get unloaded from the blood because its partial pressure is higher in the blood. And carbon dioxide will be loaded in because its partial pressures are higher in the tissues. So it gets loaded into the blood. Any questions about this?
No.
So this is just kind of a different depiction of that same idea. Oxygen is going to, everything always flows from a higher partial pressure or higher concentration to a lower concentration. And so in the systemic capillaries, the partial pressure of oxygen is 100 millimeters of mercury. The partial pressure of carbon dioxide is 40. In the tissues, in comparison to the systemic capillaries that are coming from the lungs, that blood that's coming from the lungs, the tissue cells have a partial pressure of oxygen of 40. And the carbon dioxide has a partial pressure of 45. So carbon dioxide is going to flow into the blood because its partial pressure is lower in the blood. And the oxygen is going to flow into the tissues because its partial pressure is higher in the blood. So like all things, things are just flowing from a higher concentration to a lower concentration, and that's always dictating the way things are going to move. Wherever the concentration is highest, it's going to move from there down to where it's a lower concentration. One other thing that we keep in mind here is that there is a ventilation perfusion coupling, and I'm going to come back to the whiteboard for a minute. Ventilation is airflow. Ventilation is going to be the rate of airflow in the alveoli. Perfusion is going to be the rate of blood flow in the capillaries surrounding the alveoli. These rates need to be the same. I'm trying to remember, I think ventilation is called Q many times, and perfusion is called R. Thus, Q, the ratio of Q to R we want it to be 1. We want it to be 1. So the rate of the airflow needs to equal the rate of perfusion. Ventilation perfusion coupling is done in order to maintain this ratio and keep them equal to 1. So, for example, we get decreased airflow, meaning that our ventilation goes down, our Q goes down. The result is that there is going to be less oxygen in the blood vessels, because if there's less airflow, there's less oxygen. What that means is that ventilation has gone up, but our perfusion is the same. We need to make them equal again. So what happens is vasoconstriction of the pulmonary vessels. If you vasoconstrict the pulmonary vessels, then we decrease the blood flow, and the decreased blood flow equals the decreased airflow. And then the ratio is at 1 again. Increased airflow. So now ventilation is up. Results in elevated partial pressures of oxygen in the blood vessels. But perfusion is still where it was before. It's still fairly low. And so so to match them again, remember ventilation needs to equal perfusion. Ventilation is airflow, perfusion is blood flow. To match them again, the blood pulmonary vessels are going to dilate to increase the blood flow, so the blood flow equals the airflow. Is this clear? Okay.
You said the blood flow equals the airflow, right?
Yes, it needs to. And so we're going to alter them. We're going to adjust them. We're either going to constrict or dilate the bronchioles, or we're going to vasoconstrict or vasodilate the arterioles in order to get them to equal. Is that clear, Becky?
Yeah, I just wanted to double check. Thank you.
You're welcome. So now, if for example, now we have decreased blood flow, so now it's the perfusion that's dropped. If we have decreased blood flow, we have reduced partial pressure of carbon dioxide in the alveoli because the blood isn't bringing in carbon dioxide. Therefore, we have reduced perfusion. We need to have reduced ventilation also. So in response, the bronchioles are going to constrict, decreasing airflow. And the result is that the airflow will match the blood flow again. Well, what if the vessels we have increased perfusion? Increased perfusion of the blood is going to result in elevated levels of carbon dioxide in the alveoli. Which means that our ventilation is higher than our perfusion again. To equalize them out, the bronchioles are going to vasodilate, resulting in increased airflow, which will match the increased blood flow. So we're either going to constrict or dilate the vessels, or constrict or dilate the bronchioles in order for them to match. Which law states that the total pressure of a gas mixture is equal to the sum of the partial pressures of its individual gases?
A.
A,
Or
C.
C.
it's
C.
C. It's C.
Dalton.
It's C. We will, Boyle's law, what Boyle's law had to do with the fact that if you increase the volume of an area, a compartment like your lungs, you decrease the pressure. Henry's law had to do with the fact that gases will move according to their own partial pressures to equilibrate in liquids. Factors that affect the diffusions of gases through the respiratory membrane. Membrane thickness. If you think about some of these, a few of these are pretty intuitive. Because if you think about What do you know about diffusion in general?
Goes from a higher concentration gradient to a lower.
Yep. And how does it change over distance? How efficient is diffusion over distance?
It's less pressure.
Let me say it differently here. I have this paper right here. And this paper right here. How hard is it going to be for something to diffuse from this side of the paper to this side of the paper?
Well,
Not hard.
Not very hard. Not very hard. It's not very thick, right? The paper. How is that going to change with regards to this?
Much harder.
Isn't
it's gonna take
It-
longer.
It's going to be much harder. So membrane thickness, the thicker the membrane, the less diffusion you're going to have. It's not going to work as well. What if this is a fairly porous thickness? How easily is it going to diffuse through?
Easier.
It's going to diffuse fairly easily. What if it's not very porous? What if it's like still, or like 2 inches of still?
It'll be more difficult.
It's going to be more difficult. If, let's say, actually, so when we're looking at membrane thickness, thicker membranes are going to result in lower diffusion rates. The diffusion coefficient, how easily does a gas move through a liquid or a tissue? If it diffuses easier, it's going to exchange a lot easier. Carbon dioxide is 20 times more diffusible than oxygen is. Surface area. The more surface area you have, just like we have with cells, you know how they like to maximize their surface area with regards to volume, because that gives them more area to diffuse. Same thing here. The increased surface area is going to result in increased diffusion. Diseases like emphysema and lung cancer reduce surface area.
yes.
Yep. Partial pressures. Gases move from an area of a higher partial pressure to an area of a lower partial pressure. Normally, the partial pressure of oxygen is higher in the alveoli than in the blood. The opposite is true for carbon dioxide. So, factors affecting diffusion of gases: membrane thickness. How easily does that gas diffuse, that particular gas? What's the surface area like? And what are the partial pressures? Which of the following would slow down gas exchange between the blood and the alveoli?
I could be wrong, but B.
B. You're close, Jade. Will increase surface area, will it, if something is increased in its surface area, will it allow for better diffusion or less diffusion?
Better.
It allows
Better.
for better.
Better.
B.
So,
I want to
It'll be-
say A.
A.
I would say A2. So looking at just oxygen and carbon dioxide, because that's what we really care about when we're talking about the respiratory system. Oxygen moves from the alveoli into the blood. Carbon dioxide is going to move from the tissues into the tissue capillaries, aka the blood. The blood is almost completely saturated with oxygen when it leaves the capillaries. Carbon dioxide is going to move from the pulmonary capillaries into the alveoli. The partial pressure of oxygen in the blood decreases because of the mixing with deoxygenated blood. And oxygen is going to move from the tissue, from the blood into the tissues. In a healthy person, which one will have the greatest influence? Which one will have the greatest influence on resistance to pulmonary airflow?
C.
How did you know that?
Was on the test.
Oh, you know what, Becky, how else did you know that from what we've just been talking about? What, let me put this a different way. Let's look at this air as being fluid. What do you know from when we talked about blood vessels? What has the most effect on the flow of the blood?
The diameter of the vessels.
Right.
So be the same thing. The diameter.
Exactly. It's the same thing here. Yes, you're right. It was probably on the quiz too, because guess where I get these questions? That's awesome. Okay. Oxygen is transported by hemoglobin, and the saturation is about 98.5%. And it is dissolved in the plasma, but it's not very soluble in the plasma. And therefore, its percentage in the plasma is about 1.5%. The oxygen hemoglobin dissociation curve describes the percentage of hemoglobin saturated with oxygen at any given partial pressure. You guys, how many oxygen combined to one hemoglobin molecule?
4.
Good job, 4. You guys are smart. And let's see, what was I going to say next? The oxygen hemoglobin dissociation curve shows that at rest, hemoglobin is almost completely saturated if the partial pressure of oxygen is over 80 millimeters of mercury or above. At lower partial pressures, the hemoglobin will release the oxygen. Now, you will learn, I've got some figures, and these are not in your books. I use a lot of figures from a lot of different things, but I think I told you maybe a week ago, maybe a little longer, that I told you this was my first time teaching biology 122, or the second part of A&P. And I kind of fibbed with that one. I didn't fib, I had my first time ever teaching the second part of A&P was over a summer. And it was a 5-week course, and it was 4 days a week, 4 hours a day. And I had about a week to prepare before it started. Oh, it was nuts. I think it's been a long time since I have not cried that much teaching since then, because that was really, really hard for me. But all the students survived, and so did I, and nobody died, and life was good. But I can tell you that I remember very little about what I taught during that time and what subjects we covered, and because it was all blurred to me. But I was trying to figure out, how do I cut my prep time down and make my lectures better for this class? And not have it be a better class and do the right thing by you guys. And I went back and started looking at those PowerPoints from that class, which was 2 years ago, 2.5 years ago. And realized, some of their figures were better than what I had. And some of my explanations, even though I don't remember a whole lot about what happened over that 6-week period, my PowerPoints were not as bad as I thought they were. And so a lot of these figures are coming from those PowerPoints now, because I'm going back and reusing a lot of that material. And I hope it's better for you. You can give me feedback and tell me what you think, comparing this PowerPoint to the other PowerPoints you've had so far. And you can tell me what you think. But this is one of those figures. Go ahead, say it again.
I learned a lot in your lectures, so these are great.
Okay, well, this is one of those figures that came from this new PowerPoint, this old PowerPoint. When I realized they weren't as bad as I thought they were, even though I could barely remember them, they weren't as bad as I thought they were. So this oxygen, what this shows is that as oxygen binds to hemoglobin, remember you can bind 4, so each of these circles here are showing one of the heme groups within the one hemoglobin molecule. So looking at this graph, partial pressures of oxygen in the blood with respect to the percent saturations of oxygen in the hemoglobin. When the first hemoglobin is very hard to bind to the first oxygen is very hard to bind to the hemoglobin molecule. After it binds, the second one is easier to bind. The third one is easier to bind than the first 2 were. However, then getting the fourth one on is harder. And removing that fourth one is harder. But once the first one comes off, gets removed, the second one gets removed easier. The third one gets removed easier. And we're going to watch a video here explaining this a little differently. So oxygen hemoglobin dissociation curve. So actually, before I show this video,
And today,
stop. Before I show this video, understand that the oxygen hemoglobin dissociation curve is mainly is indicating how oxygen binds to hemoglobin and how easily or hard it's removed. How hard is it to remove it, or how easy is it to bind it?
In this video, we will be talking about partial pressures when breathing and show you how to read the oxygen and carbon dioxide dissociation curves. This will be a continuation of part 1 of this segment about gas exchange, where we talked about hemoglobin and diffusion. As we explained in part 1, we breathe in oxygen gas and breathe out carbon dioxide gas. Gas exchange occurs in the alveoli, microscopic hollow sacs at the ends of the bronchioles in our lungs. Our atmosphere is made up of several gases, including oxygen and carbon dioxide. Each individual gas exerts a partial pressure that adds up together to produce the total pressure of our atmosphere. Partial
Remember, that's Dalton's law.
pressure is a measure of how much of a gas is present. For example, in our bodies, there's more oxygen in our lungs as we breathe in, which means it exerts a higher partial pressure. There's less carbon dioxide, and thus it has a lower partial pressure. We can plot the changes in the partial pressure of oxygen in our blood on an oxygen dissociation curve. This graph is demonstrating the amount of oxygen that binds to hemoglobin in the blood at varying pressures of oxygen. On the x-axis, we have the different pressures of oxygen. The y-axis is the percentage of oxygen saturated with hemoglobin. As you can see, the curve is sigmoidal in shape, which is due to the way hemoglobin binds to oxygen. When oxygen binds to hemoglobin, the hemoglobin protein structure changes in shape in order to attract more oxygen molecules to bind to it. You can see that there's a steep increase in the curve as the levels of oxygen increase. This is due to the structural changes in hemoglobin that makes it easier to bind to oxygen after the first molecule of oxygen is bound.
So after the first molecule binds, the others, the second and third ones, are going to bind easier. Pulling the first oxygen molecule off the hemoglobin is the hardest. But after you pull the first one off, the second one will come off easier.
Your deoxygenated blood is carried through your veins and back into your heart and lungs. In your veins, hemoglobin is almost 80% saturated with oxygen. After your blood has gone through the lungs to pick up oxygen molecules, your arteries carry blood that is almost 100% oxygenated to your tissues to supply them with the oxygen that they need. The amount of oxygen that is unloaded to your tissues is indicated by the red bracket, while the purple brackets indicate the amount of oxygen that is used.
So at about 40%, partial pressure of 40% of oxygen, remember that's about what it is in the tissues. You're at about 75% saturation. At a partial pressure of 100, which is what the oxygen level is like in the capillaries in the lungs, you're at about 100% saturation.
Usually in your veins, which carry deoxygenated blood. Oxygen binding to hemoglobin is very dependent on many factors within our body. One major factor is the acidity levels within our blood, which is in response to carbon dioxide levels. A decrease in pH levels, which means an increase in acidity, and the impact on oxygen dissociation.
How would increased CO2 levels result in increased acidity? Do you remember that chemical equation? Carbon dioxide plus water forms.
Can we take a picture of it to remember it?
Let me write it. Let me write it for you just for a quick moment here. Carbon dioxide plus water is going to become carbonic acid. Which immediately disassociates into bicarbonate a really great base, plus hydrogen ions. And since we know that pH equals the negative log of that was one of it. That's okay. So as carbon dioxide goes up, carbonic acid goes up. As carbonic acid goes up, it immediately dissociates into bicarbonate and hydrogen ions. pH is a measure of the hydrogen ion concentration. So as the hydrogen ion concentration goes up, the pH, because remember the pH, the value for pH is inversely proportional to the hydrogen ion concentration. So as the hydrogen ion concentration goes up, the acidity goes up. So as carbon dioxide goes up, the acidity is going to go up. Do you see that?
Yes.
Now, come back to this video.
Is known as the Bohr
Oh,
effect.
I don't think you can see it yet. Just a sec.
Nope, just you.
Yep, I thought, shoot, no, I knew you couldn't.
During activities such as exercise, our CO2 levels increase in the body, which produces
So CO2 levels are increasing, which means that acidity is going up.
increased levels of acidity and a decreased pH level. High carbon dioxide stimulates oxygen to be released from hemoglobin, referred to as oxygen dumping, thus our PO2 is lower. This inverse relationship results in a curve shift to the right. Now that we have explained the oxygen dissociation curve, let's look at the dissociation curve for carbon dioxide. The y-axis on the carbon dioxide dissociation curve shows the amount of carbon dioxide that is absorbed by the blood. The x-axis on the carbon dioxide dissociation curve indicates the pressure of carbon dioxide. The reason the carbon dioxide dissociation curve is hyperbolic is because at lower temperature pressures, more carbon dioxide is absorbed by the blood. Eventually, as we can see, as the pressure of carbon dioxide increases, the volume absorbed stays slightly linear. This is because the blood can only absorb a given amount of carbon dioxide. Therefore, at lower pressures, it absorbs as much as possible, but as the pressure increases, it decreases its absorption slightly. The Haldane effect is a property of hemoglobin and occurs when deoxygenated blood is able to carry more carbon dioxide at any given pressure, as shown in curve A. Conversely, oxygenated blood has a reduced capacity for carbon dioxide, as shown in curve B. The effect is large enough to account for much of the carbon dioxide leaving blood as it flows from the venous to arterial side of the lungs. As seen in the image, deoxygenated hemoglobin binds carbon dioxide molecules and therefore picks up the carbon dioxide molecules in the tissues more readily. Then in the lungs, the Haldane effect promotes the dissociation of carbon dioxide in the presence of oxygen, and the hemoglobin is free to load oxygen molecules. Let's recap what we learned today. Partial pressure measures the amount of gas present. The oxygen dissociation curve demonstrates binding of oxygen to hemoglobin. This curve shifts to the right when there's higher levels of carbon dioxide present, which is referred to.
The Bohr effect is the effect of pH, meaning increased carbon dioxide levels on oxygen binding. We'll see that in a second also.
As the Bohr effect. The carbon dioxide dissociation curve demonstrates binding carbon dioxide to hemoglobin. This curve shifts up when there is higher levels of oxygen present, which is referred to as the Haldane effect. Meaning
that when there's higher levels of carbon dioxide, oxygen is going to be more easily released from the hemoglobin. When there's higher carbon dioxide levels, oxygen is going to be easier released, is going to be released a lot quicker and easier from the hemoglobin. Write that one down. Because it means everything. When, let me say it one more time. When carbon dioxide levels are high, oxygen is going to be easily released from the hemoglobin or be released quicker. We'll end this video and come back to what we were looking at. So at point A, where we're at a partial pressure of oxygen of 100 millimeters of mercury, the hemoglobin is almost 100% saturated with oxygen. At 40 millimeters of mercury, hemoglobin will only unload about 25% of its oxygen. Therefore, in venous blood that has an average partial pressure of oxygen of 40 millimeters of mercury, the hemoglobin is about 75% saturated. If a person exercises really vigorously, the hemoglobin will unload about most of its oxygen into the tissues. In such cases, it will have only a saturation after, when it's leaving those tissues, of about 25%, or one oxygen bound to the hemoglobin. So the graph is going to display two measurements. It's going to display the amount of oxygen bound to the hemoglobin as a function of the partial pressure of oxygen in the blood and the tissues. The shape of the curve tells how the percent saturation and partial pressure of oxygen in the blood changes with respect to one another. So the percent saturation at very high levels with the partial pressure of oxygen being really high, the percent saturation is going to change very little when the pressure drops from 100 millimeters of mercury to 80 millimeters of mercury. Even when the oxygen levels in the arterial blood are much lower than normal, hemoglobin will hold on to most of the oxygen molecules. In some individuals, like those people living at high altitudes or with pulmonary disease, the arterial partial pressure of oxygen never gets above 85 to 80. Meaning that their hemoglobin are still very are at least 80 to 90% saturated. Percent saturation is going to drop rapidly when the partial pressures of oxygen are low. When it's about 40 millimeters of mercury, the partial pressure of oxygen in the capillary beds is going to drop extremely rapidly. It's going to be easily released, allowing hemoglobin to unload the oxygen where it's needed in the capillary beds. And to do so quickly to ensure that the cells are receiving enough oxygen.
I have a question.
Oh, please.
Is there a negative effect to those people that live in those high altitude places then if their oxygen level is like
The one thing that
constantly-
they do, the one thing that they have to make up for the fact that they never have those high saturation levels like we tend to have, is they have a higher hematocrit. Zena, what's a hematocrit?
Can you hear me?
Nope, now I can. What's a hematocrit?
Hematocrit is, wait, hemoglobin is the oxygen red blood cells. Okay, so the hematocrit. Isn't it, how do I explain it? So, wait, I'm trying to find out that word. Oh gosh, we learned at the beginning of the semester and I'm trying to come up with the word,
What?
but I can't think of it.
What does it measure?
Oxygen level.
No, 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 no. You're close. If people have,
Oh, the red blood cell.
yes, yes. So what does a hematocrit measure?
Oxygen, right?
Well, it measures oxygen kind of, but how is it measuring, how is it estimating how much oxygen is possibly present, or how is it doing that?
Based on the amount of like red blood cells that you have.
Right. So people who live at those higher altitudes where their oxygen saturation of their blood may never get to above 80 or 90 have more red blood cells to allow for the fact that they're going to have reduced oxygen saturation levels.
Oh, okay. So it adjusts itself then.
Yeah, I
doesn't
mean,
affect them because-
their body works to compensate for it.
Okay, that makes sense.
That's
Thank you.
well, and that was a great question. Good question. So 4 factors that are going to change the affinity of hemoglobin for oxygen by altering hemoglobin shape. The way that the oxygen gets bound up by hemoglobin or gets released from hemoglobin results in shape changes to the hemoglobin. Just like other enzymes and proteins, if we change their shape, we change their function. And so we're going to gradually change the shape of hemoglobin, which is going to allow it to either have a higher affinity for oxygen or decreased affinity for oxygen. Increasing or decreasing the temperature is going to alter hemoglobin's affinity for oxygen. More heat is generated in tissues undergoing a higher metabolic activity, which is going to decrease hemoglobin's affinity for oxygen. So, and no, I'm not going to show you my big muscles that I don't have But I have my arm here, stretch, working out this muscle, working out this muscle, working out this muscle really, 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 really hard. And what is happening to this muscle?
It's pumping blood.
It is hopefully pumping blood. What's happening to the CO2 levels within this muscle?
Wouldn't it be contrasting? I could be a little off, but wouldn't it be like slower? I mean, more pumped up, like from having that movement of the muscle, like.
I was gonna say the CO2 is you're burning oxygen, so the CO2 level would be higher.
It's going up. What's happening to the temperature?
It's going up.
It's going up. What's happening? What's happening to the acidity?
Going up.
Or it's becoming more acidic, right?
Right.
So all of these things, all three of those factors are going to allow for the hemoglobin in that area to give up the oxygen easier. So more heat is going to result in the hemoglobin having a decreased affinity for oxygen. Another thing that we have here is this 2,3-bis phosphoglycerate, which is produced as a side effect of glycolysis. When do you have glycolysis? When do you have glycolysis? Glycolysis occurs when we're running out of oxygen in the tissue because it's an anaerobic form of forming ATP. So if its levels are going up, it's because we need more oxygen in that tissue. So if there's more anaerobic development of ATP going on because of glycolysis, we're also going to have reduced affinity. We're going to have oxygen being more easily unloaded from the hemoglobin. The Bohr effect has to do with that partial pressure of carbon dioxide and acidity levels going up, which is going to allow hemoglobin to more easily give up the oxygen. So temperature, this anaerobic working out, because there's not enough oxygen around, is going to form this 2,3-bis phosphoglycerate that when it binds to the hemoglobin is more likely to give up the oxygen. Bohr effect has increased acidity, or the partial pressures of carbon dioxide increasing, are going to result in hemoglobin being more likely to give up the oxygen. So an increase in the carbon dioxide levels causes a decrease in the pH. Carbonic anhydrase that catalyzes this reaction right here causes CO2 and water to combine irreversibly and form carbonic acid, which ionizes to form hydrogen ions and bicarbonate. Increased temperature also decreases the affinity for oxygen to remain bound to hemoglobin. So as the metabolism goes up in the muscle, more oxygen will be released. as the muscle gets worked out more, the hemoglobin in that area is more likely to give up the oxygen. This Bohr effect is the effect of pH on the hemoglobin dissociation curve. As the pH declines, becoming more acidic, the amount of oxygen bound to hemoglobin at any given partial pressure of oxygen also declines. This occurs because the decreased pH results from an increased hydrogen ion level. And the hydrogen ions bind to hemoglobin, changing its shape, making it the oxygen less likely to be able to bind to the hemoglobin. So the hydrogen ions are, as they're increasing in their concentration, they're binding to the hemoglobin, changing the structure of the hemoglobin, making it have a reduced affinity for oxygen. So when we compare this to that oxygen dissociation curve, here's the normal blood pH, 7.4. When we have elevated CO2 levels, the acidity is going down because, or excuse me, the pH is going down, meaning the muscle's becoming more acidic. And the result is that the curve shifts to the right. And the oxygen is more easily released. If the curve shifts to the left, the oxygen is held on to stronger by the hemoglobin. Questions? With increased temperatures, the hemoglobin is more likely to give up the oxygen and release the oxygen. As the temperature decreases, the hemoglobin is less likely to give up the oxygen. So a shift to the right of this dissociation curve means a reduced affinity of the hemoglobin for the oxygen. A shift to the left means that it has an increased affinity for the oxygen the hemoglobin has. So the effects of temperature, partial pressure of carbon dioxide on oxygen unloading from hemoglobin. Increased temperature causes more oxygen unloading. Remember, this means that the curve is shifting to the right. Increased hydrogen ions is going to result in more oxygen being unloaded. The curve is shifting to the right again. Increased carbon dioxide levels is going to cause more oxygen unloading. So the curve is shifting more to the right again. So when does it shift to the right and when does it shift to the left? As the pH decreases, meaning it becomes more basic, excuse me, wait, that was wrong. As the curve pH decreases, becoming more acidic, or as the partial pressure of carbon dioxide increases, or as the temperature increases, the curve shifts to the right because the hemoglobin is more likely to release the oxygen. In the lungs, as the pH increases, the partial pressure of carbon dioxide decreases, or the temperature decreases, and the curve shifts to the left, resulting in an increased ability of the hemoglobin to pick up oxygen. This would be what would be occurring in the lungs. You want the hemoglobin to easily bind up oxygen when it's in the lungs, and then be willing to give it up and release it in the tissues. Any questions? So the addition of carbon dioxide to the blood generates what in the red blood cells, which in turn stimulates red blood cells to unload more oxygen.
For some reason, I want to say potassium, but I'm not sure. I don't know why.
Come back and look at this graph right here. What's this one say?
Carbon dioxide.
Nope, nope. Look. Read the whole thing, Becky.
Carbon. How do you?
Carbon,
Anhydride?
carbon dioxide plus water.
Oh, plus water converts to, what is that? Oh. Carbon, is it carbon acid?
Carbonic
Carbonate.
acid.
Thank you. And then that changes to, I don't know what the,
Hydrogen ions.
okay, plus hydrogen.
Bicarbonate.
Okay.
Okay, so now for this question, the addition of carbon dioxide to the blood generates what in the red blood cell?
Hydrogen.
Hydrogen ions. Let's take a 5-minute break. Let's get up and move.
Thank you. I'm starting to get a headache.
You revolve.
Yeah. All right. Thank you.
Okay, let's jump back into this. Carbon dioxide transport in the blood. It's transported from the tissues back to the lungs via the blood in three ways. 7 to 10% of it will be dissolved in the plasma. Approximately 20% of it is transported by being bound to the hemoglobin polypeptide chains in the form of carbaminohemoglobin. The carbon dioxide binds to the amino groups, binds to the amino groups of the hemoglobin, forming carbaminohemoglobin. The hemoglobin that has released the oxygen binds more readily to the carbon dioxide than hemoglobin. This is that Haldane effect. So when carbon dioxide binds to hemoglobin, it reduces hemoglobin's affinity for oxygen, making it more likely to give up the oxygen. And 70%, the other 7, so 10% dissolved in plasma, 20% bound to the hemoglobin, and 70% of it is transported as bicarbonate. What is bicarbonate? Is it an acid or a base?
A base?
It's a base. So when, good job, Casey. So it's a base. So when it's vital to pH, because if you increase the amount of base, what happens to the pH?
It becomes more alkaline.
It becomes more alkaline. So that's why this is vital to pH. You guys are good. Conversion of carbon dioxide and water into carbonic acid in the erythrocytes and the bicarbonate transport in the systemic capillaries. So carbon dioxide, you guys memorize this equation. Carbon dioxide plus water equals bicarbonate plus hydrogen ions plus bicarbonate. So carbon dioxide diffuses into the erythrocytes. It's converted through its binding with water to carbonic acid. It then immediately disassociates into hydrogen ions and bicarbonate. The hydrogen ions bind to the hemoglobin. And the bicarbonate is actually moved out of the cell via what's called the chloride shift. Via a chloride shift, where bicarbonate leaves as chloride moves in. So it diffuses into the carbon dioxide diffuses into the cell. It reacts with water to form carbonic acid. Carbonic acid disassociates into bicarbonate and hydrogen ions. The hydrogen ions bind to the hemoglobin, and the bicarbonate leaves the cell as chloride moves into the cell. Most bicarbonate is going to diffuse into the plasma while hydrogen binds to hemoglobin. The hemoglobin acts as a buffer because remember that it's acting as a buffer because pH is only a measure of the free hydrogen ions. If it's bound up by hemoglobin, it's not part of the measurement for pH. Bicarbonate carries a negative charge and is counteracted by a chloride shift. As the chloride moves into the erythrocytes, the bicarbonate moves out to balance the reactions. So this is what's occurring in the interstitial tissues and the blood. The reverse reaction is going to occur in the erythrocytes in the pulmonary cavities, capillaries, sorry, not cavities, pulmonary capillaries to facilitate carbon dioxide movement into the alveoli. So the chloride shift is going to occur again, but it's going to be in the reverse. Chloride is going to move out of the cells, bicarbonate moves in. The hemoglobin that bound up all those free hydrogen ions in the tissues is going to give up that hydrogen ion, and that hydrogen ion is going to react with bicarbonate. Bicarbonate is going to be reformed in the erythrocyte. Carbonic acid. Carbonic acid then immediately disassociates into water and carbon dioxide leaves the cell. So it is the reverse of this. This is what's occurring in the interstitial tissues to carry out the carbon dioxide and take it to the lungs. This is what's occurring in the blood, in the lungs. Questions? So this is the opposite of this. This is just showing the different ways the oxygen and carbon dioxide are carried. Carbon dioxide, 7% of it is going to be carried as it's being dissolved in the plasma. 23% of it is going to be bound to the hemoglobin, making those carbaminoproteins, hemoglobin molecules. 70% of it is going to be made into bicarbonates, which will then diffuse out of the cell as chloride moves in. Oxygen, 98.5% of it is going to be transported as it's by being bound to the hemoglobin. And 0.5% of it is going to be dissolved in the plasma. Questions? When we move into alveolar gas exchange, so this is systemic gas exchange out in the tissues, systemic tissues. Alveolar gas exchange. 7% of the carbon dioxide is that dissolved carbon dioxide is going to move into the alveoli. The carbamino hemoglobin is going to break down, and the 23% of the carbon dioxide is going to flow into the alveoli. The bicarbonate that was removed from the cell in a reverse chloride shift, where now chloride is going to get moved out of the cell as that bicarbonate is going to be brought back in, is going to combine with the hydrogen ions that are coming off the hemoglobin, become carbonic acid that then easily disassociates back to carbon dioxide and water. And that carbon dioxide diffuses into the alveoli. Oxygen gets picked up here, gets bound to hemoglobin, and 1.5% of it is just going to be dissolved in the plasma. Do you have questions for me?
So like the systemic gas exchange is the opposite of the alveolar gas exchange, basically?
Yep.
Okay.
Yep.
Just wanted to make sure I was reading it right.
That's very right. Yes.
Okay.
Carbon dioxide is transported by all of the following except for what?
Is it A?
It's not, because that's what this is right here.
C.
Who said that? That was Rachel? Yes.
Yes.
So that's right. Yes. Because as far as I know, we don't have that. Effective ventilation on the ventilation pattern on blood pH. Normal pH is between 7.35 to 7.45. If the pH increases, meaning it becomes more basic, more CO2 is being expired. And if you're giving up more CO2 into the atmosphere, what's happening to your blood pH? If you're breathing out a bunch of CO2, what's happening to your blood pH?
It increases.
Becoming more basic or more acidic?
Basic.
Yes. So more CO2 being released, your pH is becoming more basic. Carbonic acid is going to be releasing less hydrogen ions, so the hydrogen ion concentration decreases, and the blood pH increases, like Becky said. Hypoventilation. Blood pH is below 7.35, so it's more acidic. Less CO2 is expired. The blood carbon dioxide level increases. Carbonic acid is going to release more hydrogen ions, because if you've got more carbon dioxide, you're going to have more carbonic acid. So it's going to give up. If you have more carbonic acid, you have more hydrogen ions. The hydrogen ion concentration is going to increase, resulting in an increased blood pH, meaning it's becoming more acidic. Which of the following is directly related to the respiratory production of carbon dioxide?
D.
D is in delta, regulation of pH. That looks right to me. Good job. Neural control of the basic pattern of ventilation. Our respiratory systems are in the pons and the medulla oblongata. The pontine or pneumotaxic respiratory group is a series of neurons found in the pons that are involved in switching from inspiration to expiration. Remember, the expiration is just the relaxation of your inspiratory muscles. So it's not actually essential to respiratory rhythm. It's just the switching is the pneumotaxic center. The relaxing of those muscles is related to the pneumotaxic center. In the medulla oblongata, the respiratory center there stimulates the diaphragm primarily for inspiration. If the diaphragm contracts, it expands the rib cage, resulting in increased lung volume, therefore decreased pressure. The ventral respiratory group stimulates the intercostal and abdominal muscles in both inspiration and expiration. If you heard recently about the death of the drummer for the Foo Fighters, I honestly was not that familiar with their band. I don't think I was ever a big fan. But opioid overdose is working on these centers. This is what they work on. So essentially what you do is stop breathing. Because it gets slower and slower and slower until you're no longer breathing at all. I learned this from experience having, so I've told you about crashing my car and breaking my jaw. And the result of one of the experiences that came with that whole adventure was I had a morphine overdose, an accidental morphine overdose while I was in the ICU. And the result is that I essentially went into a respiratory suppression and stopped breathing because of the morphine. And it works on these centers right here. So regulation of respiratory rhythm is controlled by chemoreceptors, specialized neurons that are going to respond to changes in the chemicals in a solution. Central chemoreceptors, the chemosensitive area of the medulla oblongata that's connected to these two groups right here. The peripheral chemoreceptors, which are found in the carotid and aortic bodies. Remember the carotid sinus with its receptors, and the aortic bodies, which are in the aortic arch. And these peripheral chemoreceptors are connected to the respiratory center via cranial nerves 9 and 11, or not 9 and 11, 9 and 10. Effect of pH. Chemosensitive areas of the medulla oblongata and the carotid aortic bodies are going to respond to blood pH changes. So these areas right here are going to respond to pH changes. And these areas respond indirectly to changes in the carbon dioxide level because of the change in pH. And the carotid and aortic bodies are going to respond to these changes in pH. And decrease breathing if you're acidic. And increase breathing if you're basic. Actually, it's the reverse. Increase breathing if you're acidic, breathing rate, and decrease breathing rate if you're basic, to affect the CO2 levels. So it's not oxygen that has the most popular effect on pH, it's carbon dioxide. Those peripheral chemoreceptors found in the aortic bodies and in the carotid sinuses. Any questions? So if your CO2 levels go up, your breathing rate increases because you become more acidic. As you become less acidic, your breathing rates go down because you're becoming more basic. And these breathing rates are working to adjust your partial pressures of carbon dioxide. So the role of chemoreceptors in regulating blood pH, because they're regulating blood pH through altering the rate of ventilation. The respiratory pontine group triggers hyperventilation. What is that? I don't know. Okay. So starting in one place. So partial pressure of carbon dioxide or hydrogen ion levels are increasing above their normal range. The receptors, the central chemoreceptors in the medulla oblongata and the pons are detecting this increased amount of carbon dioxide or hydrogen ion levels. The chemoreceptors relay the signal to the respiratory pattern generator in the medulla oblongata and the pons, which trigger hyperventilation so that that extra carbon dioxide that was increased is lost. And the carbon dioxide levels and hydrogen ion levels return back to normal. And the breathing rate slows. In contrast, elevated carbon dioxide levels or hydrogen ion levels are going to decrease below their normal range. So they're going to become very low, meaning the person is becoming very basic. The central and chemoreceptors detect this decrease in the amount of carbon dioxide or hydrogen ion levels. And the chemoreceptors relay this signal to the respiratory pattern generators. And they trigger hypoventilation, so you're breathing less, resulting in more carbon dioxide being saved up, accumulating in the body. And as the partial pressure of carbon dioxide or hydrogen ions return to normal, the respiratory activity will go back to the normal baseline. Questions? So regulating respiratory rhythm, the different factors that are involved. Higher centers for of the brain involving speech, emotions, voluntary control, breathing, and action potentials can affect the respiratory rhythm. Medullary chemoreceptors, these are your central chemoreceptors, are affected by pH and carbon dioxide levels. The peripheral chemoreceptors in the carotid and aortic bodies are affected by pH and carbon dioxide levels and oxygen levels. The Herring-Brewer reflex, which we haven't talked about, but has to do with increased stretch receptors in the lungs, has a negative effect on breathing rates. So it's going to work to decrease breathing rates. Proprioceptors in the muscles and joints are going to have a positive effect on respiratory rates. When your joints start really moving a lot more, your breathing rate goes up. Receptors for pain, touch, and temperature and pain stimuli are also going to cause your breathing rates to go up. And we'll see if you can answer this one. During exercise, which of the following directly increases the respiratory rates?
Is it D?
D is in Delta.
Yeah.
It might, but it's not.
Okay.
It actually is E, the anticipation of the needs of the exercising muscle.
Oh, that was on the test.
Was it on the test?
So, or something in that form. Yes.
It was somewhere in there.
Yeah, it was.
All these first three, do you see that they're all the same thing?
They're very similar, yes.
Why would I say they're all the same thing? What's the Bohr effect?
Increased CO2 levels, O2 is going to be released for quicker, easier release from the hemoglobin.
Why?
I know it, but I'm
Because,
drawing a blank.
because of the pH.
Okay.
And so, and of course, increased hydrogen ion levels are going to result in the increase becoming more acidic. Reduced blood pH, AKA you're more acidic. So some slight variations in the respiratory rhythm. Apnea is temporary cessation of breathing or skipping breaths. Dyspnea is labored or gasping breathing or shortness of breath. Hypernia is an increased rate and depth of breathing in response to exercise, pain, or other conditions. Hyperventilation is increased pulmonary ventilation and excessive metabolic demands, and is frequently associated with anxiety, expelling carbon dioxide, raising the blood pH by lowering the carbon dioxide concentration in the blood. Hypoventilation is reduced pulmonary ventilation, resulting in increased carbon dioxide levels in the blood, making the blood more acidic, and the ventilation is insufficient to expel the carbon dioxide as quickly as the carbon dioxide is being produced. So the carbon dioxide is being produced faster than you can expel it and get rid of it. Cosmal respiration is deep, rapid breathing, often induced by acidosis, seen in diabetes mellitus. Orthopnea is dyspnea that occurs when a person is lying down or in any position other than standing or sitting erect. It's often observed in heart failure, asthma, emphysema, and other conditions. Respiratory arrest is a permanent cessation of breathing, so a permanent stopping of breathing. Tachypnea is accelerated breathing. Accelerated respiration. Any questions about the respiratory system? Let's jump into the urinary system. So we're talking a lot about acid-base balance. And acid-base balance is being regulated by both the respiratory system and the urinary system. So they're working hand in hand. My goal is to finish the urinary system today. Your second exam will open up tonight. And it will cover the endocrine system, the respiratory system, and the urinary system. Also, I'm going to put your next case study up. I will start grading the first case studies over the weekend, but I will put up your second case study. I may put up your next one. I just need to think about it. Your third one. And one other thing, your LabQuest 4 is open for you.
Are you
You're
trying
giving
to
me
kill us?
anxiety.
I was like, are you trying to kill us?
Like I have anxiety now.
We have one class a week. Come on.
I guess we'll survive.
I know, I understand though. I understand.
Are we having class the Friday before Easter, which is next Friday?
Yes.
Okay. Just wanted to double check. My husband's like, no, you're not. I was like, yeah, we will. Welcome to the real world, babe.
I do believe in Good Friday, but no, we don't have it off.
I didn't think so. I just wanted to confirm so I could tell him that for sure.
No, we will.
Okay.
Urinary system functions and structures. The major urinary system functions: excreting and removal of waste, so detoxification and urine production, fluid and electrolyte balance, blood solute concentrations. So this is fluid and electrolyte balances, meaning it's regulating blood solute concentrations. Also, maintenance of blood pressure, so blood volume and peripheral resistance. Erythropoiesis. What's erythropoiesis? Guys, what's erythropoiesis? This is what gets stimulated when you live in higher altitudes and your oxygen saturation levels are never very high. Somebody help me, what's erythropoiesis?
Red blood cells production.
Yes, good job. pH homeostasis of the blood plasma, so regulating the amounts of hydrogen ions and bicarbonate concentrations. I'm gonna stop just for one quick second here. This graph right here, that is our no, sorry, not graph, our major equation for measuring blood pHs. Carbon dioxide water equals carbonic acid, that then dissociates into bicarbonate and hydrogen ions. These two things on this equation, actually not even both of those things. In order to regulate pH, the respiratory system works on this. It can alter the pH by changing the amounts of carbon dioxide. That's how the respiratory system works to affect pH. In contrast, The urinary system works on these. It can increase or decrease the levels of bicarbonate and hydrogen ions, therefore affecting pH. It can't do anything with carbon dioxide, and the respiratory system can't really directly affect bicarbonate, except to change carbon dioxide levels. When we look at respiratory acidosis or respiratory alkalosis, we're looking at CO2 levels. When we look at urinary system or metabolic acidosis or alkalosis, we're looking at these two. The urinary system can't do anything about changing how much carbon dioxide gas you have in your body. But it can indirectly affect it by deciding how much of this you keep in the body and how much of this you have in the body. The respiratory system by itself and directly can't do anything about how much bicarbonate you have in your body and how many hydrogen ions you have in your body. But it can indirectly affect them by affecting how much carbon dioxide you have in the body. Keep that in mind as we go through this. And we'll go up until 10:40, and then we'll take our hour and 20-minute break and come back to this. So maintenance of blood pressure, blood volume, pH homeostasis of the blood plasma, and vitamin D synthesis. Remember, the final step in vitamin D synthesis occurs in the kidneys. Looking at the internal anatomy of the kidney, you have the renal cortex, which has little indentations into the medulla in the renal columns that flank the pyramids. And each lobule of the kidney includes part of a column and a pyramid, parts of the flanking columns and a pyramid. The renal papilla drain the urine into the minor calyces that merge to form the major calyces. And the major calyces drain into the renal pelvis, which becomes the ureter as it exits the kidney. The blood flow coming into the kidneys is supplied by the renal artery. The renal vein is draining the kidneys. So a renal pyramid voids or excretes the urine into the what?
A.
Good job. A, minor calyx. Blood supply. Kidneys have a lot of blood. They filter a lot of blood. They receive about 1/4 of the total cardiac output per minute. So about 1.2 liters of blood per minute. That's a lot of blood if you think about it, because you only have 5 liters approximately. So from the heart, the blood moves into the aorta. In the abdominal aorta, there's a branch of that branches off into the renal artery. Renal artery becomes a segmental artery. As the segmental artery moves up in between the pyramids, it becomes the interlobar artery. As it arcs above the pyramid, it becomes the arcuate artery. Which branches off into the cortical radiate arteries, or the interlobular arteries is the other name for them. The interlobular or cortical radiate arteries are going to become the afferent arterioles, which are going to drain into the glomerulus. The glomerulus blood, whatever blood is left that doesn't get filtered, the fluid that doesn't get filtered out in the glomerulus into the nephron, is going to be received by the efferent arteriole, which is going to divide off into peritubular capillaries and vasorectal capillaries. These sets of capillaries are going to become cortical radiate capillaries. Cortical radiate veins. Cortical radiate veins become arcuate veins. Arcuate veins become interlobar veins. Interlobar veins are going to merge to form renal veins, and that will drain into the inferior vena cava. Looking at the major functional unit of the kidney, the nephron, have the renal corpuscle, which has an afferent arteriole and an efferent arteriole. Remember, afferent meaning incoming, efferent meaning outgoing. So the afferent arteriole is supplying blood to the glomerulus. The efferent arteriole is draining it. The glomerulus is surrounded by is within the renal corpuscle. And the filtrate that enters the renal corpuscle is going to move into the proximal convoluted tubule. Convoluted because it's really loopy, and proximal because it's right next to the glomerulus. From there, the filtrate is going to move down the descending limb of the loop of Henle or the nephron loop, and then move up the ascending limb or loop of the loop of Henle, and enter the distal convoluted tubule. One thing that you want to keep in mind about this drawing is that the distal convoluted tubule is right next and it abuts right up against the arterioles that are supplying the glomerulus. This is important later. The filtrate, as it moves to the distal convoluted tubule, is going to enter a collecting duct and drain into the collecting duct, which will ultimately drain into a renal papilla, which will drain into renal papilla, drain into the minor calyx. We have two types of nephrons. We have cortical nephrons and juxtamedullary nephrons. The major difference between these two types of nephrons is how long are their nephron loops, or their loops of Henle. The juxtamedullary nephrons have very, very long loops that extend deep into the medulla and are surrounded by the vasa recta. The juxtamedullary nephrons, their loops go very slightly into the medulla, but they don't extend down deep. Most of the nephrons in your kidneys are cortical nephrons. About 20% of them are juxtamedullary nephrons. Any questions so far? Kidney function is a series of filtration, reabsorption, secretion, and water conservation. Whatever is in the kidneys after we extend through all four of these processes and we move all the way through the nephron, whatever is left right here that's still in the nephron is going to be excreted as urine. So it's going to be important later. Whatever is still in or is present in the filtrate down here is going to be removed from the body as urine. So 4 major functions: filtration, creating a plasma-like filtrate of the blood in the glomerulus. Tubular reabsorption, many of the different solutes that are present in the filtrate will be returned to the blood. Tubular secretion. Many of additional wastes that were filtered out will be added to the filtrate from the blood. Water conservation. Much of the water that's found in the filtrate will be conserved and returned back to the blood, concentrating waste. So filtration is going to produce a filtrate where smaller substances like water and other solutes are going to enter the glomerulus. Reabsorption. The majority of the water and other solutes within this filtrate, it's in this tube, is going to get reabsorbed and returned to the blood. And this is going to occur in the proximal convoluted tubules and the nephron loop. Other solutes, depending on what's needed in the body at any given moment, is going to be reabsorbed in the distal convoluted tubule and collecting duct. This is going to be more, the reabsorption that occurs in the proximal loop and nephron is going to be not very specific. It's not going to be as specific. Most of what will get reabsorbed here. In the distal convoluted tubule and the collecting duct, depending on the hormones available and the conditions within the body, certain things will be reabsorbed while other things won't. The nephron is going to vary the amounts of different substances reabsorbed in order to maintain homeostasis as the body's needs change. Tubular secretion. Along the entire tubule, so proximal convoluted tubule loop of Henle, or nephron loop, and distal convoluted tubule and collecting ducts. Paratubular capillaries, paratubular meaning they're surrounding the tubule, are going to add substances to the filtrate, which will then be excreted. Some things are secreted more from the tubular region than others in order to maintain electrolyte and acid-base balance. And toxins will be removed from the blood that were not filtered out initially in the glomerulus. Looking at the glomerulus means a ball of yarn. It looks like a major ball of capillary strings. We have a simple squamous epithelium that's surrounded by smooth muscle foot processes called podocytes. Then there's a small space, the capsular space, and the capsule is lined with simple squamous epithelium, which will then drain into the proximal convoluted tubule. So paratubular so glomerular capillaries surrounding the tubules are the paratubular capillaries. An afferent arteriole that's bringing the blood into the glomerulus, and the efferent arteriole that's taking blood out. The blood is going to be filtered here and filtrate will be produced. You guys, this sounds like a really great place to stop. I will see you in an hour and 20 minutes, and we will continue from here and finish the urinary system.
Sounds good. Thank you.
Was the urinalysis, I sent you another email about one of those, the Wiley labs. Was that ever opened? Because I don't think I ever saw it open.
It should have opened. I thought some students.
Yeah, I'll need that open too. I didn't see it either.
Yeah,
thought
I
it
didn't
was-
either.
It wasn't just me. Okay.
All right, well, let me